Hi, this is Miss Litton, and this is my AP Biology 7th period review of the Unit 4 essays. Yay! Before I start, could you tell the person next to you, have you even looked at them? Go ahead, have that discussion. All right. So could you think of some reasons why it would be beneficial to look at them before you come to this review? Who can come up with a good reason? You know what questions to ask. Very good. If you would wrestle with it a little bit before you came here, that would be super helpful. Okay, better for you. Okay, so the first one, I believe, is concerning the control of gene expression in prokaryotes. Hang on one second. Hey, Jacob, I'm in the middle of class. Do you need me, baby? Oh, they should be in the basket by the front door where she put them. I haven't taken them from that basket. I never took the car and moved the car, so it should still be in that basket. Okay, I love you, sweetie. I'll call you in just a little bit. Okay, bye. Sorry for that interruption. Sorry. Okay, I'm glad we have that recorded for all time. Okay, concerning the control of gene expression in prokaryotes, what is an operon? What is an operon? Anybody? Where is an operon located? On the DNA? Yep. Yes, okay. So an operon is a region, a control region, and those genes that it controls, the genes that it is in charge of. That would include polymerase binds to the promoter and operator and the structural genes. Right? Those items. Okay, so B, you would tell me what are the genes on the operon and what does each do? What's the promoter? Where RNA polymerase attaches, right? And then what's the operator? A repressor could attach, and the structural genes are whatever you're coding for, which could be a series of different proteins. Okay, something outside of the operon but associated with it is the enhancer. Okay, that would be more for eukaryotic control, and I agree, but this is prokaryotes. Okay, I'm looking for something else. You told me about something in there. Yes, the regulator. Yeah, the regulator genes sit outside of the operon usually, but they code for the repressor, and so you probably wouldn't want to address that there. Okay, then it says, what is the difference between an inducible operon and a repressible operon? So, an inducible operon is it's in the starting position. Let's do inducible and repressible. Okay, in the starting position of an inducible operon, is it on or off? Is the operon considered on or off? Off, because it's inducible, right? So the operon is off, okay, when you first start off. Okay, so if it's inducible and it's off, and it could be a couple of different things here, right? We looked at the trip, if you remember that, tryptophan, and we looked at the lac operon. Do you remember that? Okay, so we'll address both of those. A repressible operon, is it on or off? On. It's on. So it is repressible, right? Because it's on. If it's repressible, usually you need something to turn it off. So it starts off here, you're going to end up here. What do you need to transition it to off? You need a what? A co-repressor. Yeah, a co-repressor, which in our example, in the trip operon was tryptophan. And tryptophan would take an inactive repressor and make it active. Right? Okay. If it is an inducible operon, it starts off, and you want to turn it what? On. In order to turn it on, you have to remove what? You have to remove the repressor. What do you use to remove the repressor with? an inducer, and in our example, that was lactose. Does that make sense to you? Okay, both of these, because you're dealing with a repressor, either removing a repressor or you're activating with a co-repressor, both of these are referred to as negative control. Negative because you're dealing with a repressor. Positive control is what you see most in eukaryotes. You usually see mostly positive control. But we saw positive control even in prokaryotes. What was the example of positive control with our prokaryotes? In the absence of glucose, you have more what? Cyclic AMP. Cyclic AMP will bind to CAP. CAP is a transcription factor that helps RNA polymerase bind to the lac operon. You want me to say that again?
Yes.
I don't know that I'll say it the same way because I was making it up as I went along. Okay, so these are examples of negative control. In positive control, if I wanted to talk about that, let me get a new page here. For positive control, you're usually looking at a transcription factor. And the example that we discussed is when you don't have glucose, then you will see an increase in cyclic AMP. Cyclic AMP binds to CAP, and CAP is a TF for RNA polymerase and the lac operon. There, how's that? Good? There we go. Yes?
And the cap is like right before the operator.
It has to be near the promoter, usually. But we saw examples where you have an enhancer and an activator, right? That's more in eukaryotes, where you can have several different...
Brennan Fowler, please report to the vehicle. Brennan Fowler, please report to your vehicle. Thank you.
Yes, she's probably, report to the vehicle now. We're leaving. Yeah. That's funny. Okay, so yeah, that would be more another example. But if you're going to help RNA polymerase bind to the promoter, the DNA could loop back down around and then help it adhere. In the lac operon, it's right close to it. It binds, helps open up the DNA a little bit, so it makes it easier for RNA polymerase to bind. Okay, I believe that addresses A, B, and C. We good? All right. So the next question you had is concerning the control of gene expression in eukaryotes. List and describe the 5 levels of regulation and state whether the regulation occurs in the nucleus or in the cytoplasm. So let's do that. So nucleus. Nucleus. What kind of control happens in the nucleus? What's the first level? Before transcriptional control. You can talk about epigenetics, right? And you could talk about chromatin. And you could talk about the supercoiling. And I believe in your notes I said chromatin remodeling complex, something to unwind and make the DNA even available. Remember that? That was the first part when we talked about epigenetics. If it's methylated or not, whether it can come off of the histone protein. That's your first level. What's your second level? Transcriptional control. Okay, so this is going to be anything that has to do with can RNA polymerase bind to the what? Promoter. Okay, then we have post-transcriptional control. What does this have to do with? What? What's post-transcriptional control? Yeah, cap, tail, 7-methylguanosine, the tail, poly-A tail, cutting out the introns and the ability to leave the nucleus. Yes? Okay? Then we're going to get out of the nucleus and we're going to get out in the cytoplasm. And the first one we would do is what? Oh, scary. Translational control, yeah? Can RNA polymerase bind to the promoter? Or sorry, forgive me, not that. Can the mRNA bind to the ribosome, first the small, then the large ribosomal subunit? Are there small interfering RNAs? How long does the mRNA last? Is it getting digested? These are all translational control mechanisms. And number 5 is post-translational. And that has to do with chaperone proteins. Is it getting folded correctly? How long is the protein lasting? Okay, now look at me. I know you're writing a lot, but do you understand what I'm talking about so you could reference this better? Do you remember that one slide in the presentation where I listed all 5 in little summaries of all 5 control mechanisms? You might reference that, just to make sure you're in the right place. And we went through in the presentation, I think there were, a different box for each level of control. All right, anything else you want to ask me about that one? Okay. I put these two together because it made sense. And if we discuss one, we're probably discussing the other one as well. So a couple of things. Using the term plasmid. So first of all, what is a plasmid? A vector. Now tell me what it is, though. Yeah, it's usually a single circular DNA molecule, usually bonus DNA. Restriction enzymes. What do restriction enzymes do? Cut DNA at a specific series of bases called the recognition sequence. And ideally, it would cut it so that there are stick-ins, which are overhangs in the DNA, so that you can mix and match. What is DNA ligase?
Seals.
Yep, tapes it. So if you have complementary sticky ends, you can combine them, and the beauty of that is you can make new combinations. Host cell and cloning. Once you make the DNA is just a code book, like a thumb drive. You don't know what's on the thumb drive until you attach it to a what? Computer. So your DNA, the plasmid, once you've manipulated it, your computer that you're putting it into has got to be a living cell. Because what you're hoping to do is take that DNA and get it transcribed and then translated into a protein, right? So you've got to get it into a host cell. Either a bacterial cell would be the easiest of all, but there are ways to get it. Remember, we looked at transgenic organisms. We could get it into a plant. We could get it into even an animal. Okay, cloning, every time that cell replicates itself, this would be more likely in bacteria. When it makes a copy of itself, it's going to copy the plasmid or the DNA as well. And explain how biologists can produce bacterial cells that make human insulin. And I gave you several charts. I would kind of go through that. And your book itself goes step 1, step 2, step 3. If you Googled it, you would see step 1. And also, if you took a quiz today, you know about the steps, right? First, you have to find the gene. You have to cut the gene out. You have to move that gene to where you want it to go. But what you need to do on this one, though, is you have to, I would include, though this is not in this per se in this question, because you are getting it to make human insulin. Remember how we talked about how when you isolate that gene from a human cell, eukaryotic mechanisms has all that genetic junk, so you need to have a clean copy. So how would you get a clean copy?
Use reverse transcriptase.
Yeah, use reverse transcriptase from the mRNA that codes for the human insulin. You're like, oh yeah, that's all we have to do, Miss Lynn. And you need to make sure you have what kind of regions? Regulatory regions to turn the gene on and off. I'm so proud of you guys. You just spit that stuff out like you're nobody's business. Okay, so any question on that one? Yes?
I'm just a little confused with like how you use reverse transcriptase.
Oh, okay. So bacterial DNA, they don't have the junk that we have. Just making sure Michael didn't turn that off. We don't have, we have mechanisms to cut out the introns, right? But bacterial cells don't have that ability. So if we took DNA, and we have this DNA that's, the plasmid itself, and if we cut out a section and put in our human DNA in here, and let's say we have regions of it, okay, I don't know why that's up like that. We have regions of it that's junk. When we make our mRNA message from this, we know how to remove the introns. We have the ability to take it out. They don't. So what you want to do is take mature mRNA mature mRNA that's been processed, and you want to use reverse transcriptase, where it will make a DNA copy, and that's what you insert into the host, a DNA copy of the mRNA, and put that back into the host, clean, with no genetic junk, no introns.
Okay.
Does that make sense?
So
Where does reverse transcriptase come from?
Oh, it's from retrovirus.
Yes, from retrovirus. Of course it is.
Johnny!
Boom, shagalaga.
So it's like you have the DNA, then you make the mRNA, which take out the introns, and then now that's all.
No. Like, this is what I would do. If I wanted the human gene in a protein, right? My DNA has junk in it.
Yeah.
So in my cell, find the mRNA from that DNA,
And
right? The mature mRNA. Then work backwards and formulate DNA and use that formulated clean DNA and insert that into the plasmid, not the original DNA itself.
Thank you.
Okay. All right. Next, from that, any other questions on that one? Okay, let's go to the next one. Using the concepts of DNA, the concept of DNA. PCR, what is PCR? Polymerase chain reaction. So that's where you photocopy DNA. How do you photocopy that DNA? You have to what it? Heat it up, okay, to denature it. You have to put in what? Starts with a P. Primers for the regions that you want. You have to put in some what? Bases, some nucleotides, and you need an enzyme. You need an enzyme that won't be denatured in heat. TAC. Nice. Which is an archaea, right? That won't be denatured. Good. And then you can, through cycling, series of hot, cold, hot, cold, hot, cold, then you're going to be able to make multiple force replication, basically to happen exactly where you want it to be done. Using restriction enzymes, we can do what to the DNA? Cut it. And then we can do gel electrophoresis to see how big those fragments are. And you could use small tandem repeats as stirs, right? We talked about that are unique to each one of us, and you learned about those last year in Foundations, right? Or you could use other sequences of DNA and use that because each of us have parts of us that are unique, our DNA that is exactly the same. That 0.1% or whatever that's unique. And that's how you get a DNA fingerprint. Questions, concerns, issues? Okay. Okay, the next question refers to Hardy-Weinberg, and I have not taught you that. That is in our last chapter. So I think we're going to want to wait on that and then we can come back and do this question, or you do it as a review question. I don't think, is there another one?
Yeah,
this one we can talk about a little bit, okay? But this is also in the next chapter. The modern theory of genetics says that if allele frequencies change, then evolution is occurring. For instance, in a nutshell, this is kind of Hardy-Weinberg in a nutshell. How many of us in this room have blonde or red hair? Don't count me because I'm a fake. Okay, 2. Okay, 2. And how many people are in this room? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. 2 out of 18 have the recessive trait. Could you give me a percentage on that? What's 2 divided by 18?
It's 11.1%. Oh yeah? That's right.
Okay. So that recessive trait, Q, I'm pre-teaching you, is a recessive trait. P is a dominant trait. Those of you who have the recessive trait, both of your alleles are recessive. Do you agree with me on that? Now, those of us who have dark hair, we could be PP, dominant, dominant, Or we could be PQ, which would be heterozygous. You with me? Lose you yet? Okay. This would be like big T, big T. This would be like big T, and little q would be like little t. Are you all right with that? Yeah. Okay. So Q squared is 2 out of 18 people in this room, which is how many? What did you tell me? 11%? Okay. So now, you ready for this? You smart learners. Let's take the square root to solve for Q. So take 11.1 and take the square root of it. Give me a rounded number. Give me something close.
3.3.
3.3?
Yeah.
The square root, 11.1 square root. Yeah, I get 3.3. So Q is 3.3. Come back to me. Could we solve for P then? If we know Q is 3.3, then what is P? Oh, and you know what? You should be 0.33, shouldn't you?
3.3.
No, 3%.
Yeah, 1%.
It is percent, so it should be
0.33 would be the number.
0.33.
So the other one would be
Yeah, so what would this one be?
0.67.
0.67. Yes? You right? Now from this, based on if we were an interbreeding population, which we're not, but if we were, we could predict how many of us in the room were homozygous dominants for it. You ready for this? A Punnett Square. When we did Punnett Squares before, and we would separate out big T, little t, right? Cross with big T, little t. Now we can do Punnett Squares for a whole population. So you could do frequencies. Frequencies this way. So 0.67, 0.67, 0.33 right here. So P times P is P squared. P times Q, P times Q, and Q squared. P squared plus 2P squared plus 2PQ plus Q squared equals 1. These are all the homozygous recessives. These are all the heterozygotes, and these are the homozygous dominants. So I could predict, because 2 out of 18, I could predict the percentage of us that are actually homozygous dominant. Take 0.67 times 0.67, somebody, times 18.
It's 0.67.
Yeah, 0.67 squared. Good job.
0.45.
Take 0.45 times 18.
It's going to be 8.08.
8 of us are probably homozygous dominant. And then do you see, you could then solve for how many of us are homozygous, how many of us are heterozygous, and how many of us are homozygous. So that is allele frequencies. Now listen to me on this. If the allele frequency changes from 0.3 and 0.67, let's say we get even fewer blondes, okay? And let's say the Q allele frequency goes to 0.2. That would mean P goes to what?
0.8.
0.8. If the allele frequencies change, evolution has occurred. Okay? And so you use those two equations between the allele frequencies that you would find and how that plays out in the geno and phenotypes, and then you can establish whether evolution is occurring. Now, all evolution is change over time, right? And if it only changes and you're still the same species, that's called what kind of evolution? Microevolution. If we change to an entirely new species, that's called macroevolution. Yes?
I understand everything up there. I just don't understand, how'd you get the PQ and the PQ on the Punnett Square?
Oh, it's because now, instead of doing Punnett Squares where I'm crossing this with each other, I'm crossing the allele frequencies of a given trait in a whole population. So I am counting up, I could have in a population, I could have some of these, I could have some of these, maybe some more of these in my population. So I'm counting up all the big T's, I'm counting up all the little t's, and in this case, right, if I have a total number of T's, 1, 2, 3, 4, 5, 6, I have a total number of 12 alleles, right? How many of those are big T? 1, 2, 3, 4, 5, 5, right? So how many little t's should I have if I only have 12? What?
7.
7. So right here, this gives me the frequency of the P allele. So that is, because if it's not P, it's what? Q. So P plus Q equals 1, right?
Yep.
So now if I'm crossing all of them, now my Punnett square, I'm giving the frequency of the P and the Q allele, and you should recognize P times P is P squared. This is PQ, this is PQ, and this is Q squared, right? So I'm just saying P squared plus 2PQ plus Q squared equals 100% of our population. The homozygous dominants plus all the heterozygotes plus the homozygous recessives are our total population. So you can analyze that. I could give you the allele frequency and you could predict. Like if I said the allele frequency was 0.5 and 0.5, then how many would you predict are homozygous dominant? Homozygous dominant, P squared. 0.5 times 0.5 is 25. 25% would be homozygous dominant. How many would be homozygous recessive? 0.5 times 0.5 is 25. 2 times P times Q would be all the heterozygotes, and that would be 50%. So if I tell you this trait happens in 1 in 10,000 children, it's a recessive trait, you could calculate the Q squared, and you could calculate the allele frequency of that particular trait. If the allele frequencies change, what has occurred? Yeah. Where do you think it's more likely to happen? A big population or a small population? Where do you think you can make a drastic change faster? Small population. If look at how many there are of us in this room. If we're all put on an island and this is all we have to interbreed with, is this a fair representation? Is this a fair representation of the United States? Probably not. Okay? So, and the recessive trait, good luck hanging on to that, because we have some dark-haired individuals in this room. Okay? So good luck with that. Okay? So you have something called genetic drift, and that's where you have chance. So that's why you would have a large population. If a busload of kids gets brought into our little island, that's gene flow. All of a sudden, that's going to affect our allele frequency, right? That's emigration and immigration. Those are types of things. What if one of us is carrying a mutation, right? We're going to have more of that. What if there's inbreeding, associative breeding, natural selection? What if some of you are better adapted? These are things that all could happen. So what the Hardy-Weinberg principle says is, if you don't want evolution to occur, you have to meet these 5 conditions. And since it's so hard to meet those 5 conditions, then probably what occurs? Evolution, by default. That's basically the next chapter, right there. Okay, so that if you look at this essay, which now I actually did just teach you, okay, it says indicate the conditions under which allele frequencies P and Q remain constant from one generation to the next. They have to be large, there's no gene flow, there's no mutation, every mating is random. So, period 1, the 5 fingers, the hand. Period 3 didn't have time to watch it. Period 1 didn't either, but they watched it anyway. Okay. So, and then here's one you can calculate. I just did a sample one for you. You could do one yourself. And then see if the homozygous dominant condition were to become lethal, what would happen to the allelic and genotypic frequencies? If the homozygous dominant condition became lethal, what would happen? What would happen to the P allele? It would go down. Would we completely eliminate it? No. Why? It would be maintained in the heterozygote. Yes. Perfect. Okay? So now, see, we did review everything because you're so smart. So now it says describe the modern theory of evolution. So the modern theory of evolution says if you change the allele frequencies, evolution is occurring. The difference, including examples between micro and macroevolution. Microevolution is evolution within the species. I would probably discuss industrial melanism, those moths. Remember that? I talked to you about the moth and and the pollutants, and then the black allele frequency, the dark moth went up because of the pollutants on the tree. And then macroevolution is evolution into an entirely new species, okay, from another one. And then 4 categories of evidence of evolution with an example of each. Well, let's go over all the pieces of evidence of evolution. We have discussed that. What was the first piece we looked at? Fossils. Fossil has to be how old? 10,000 years old. Good. Are there transitional fossils? Yes. What would be a good example of if I was going to use fossil evidence? What was a really good one that we looked at? Oh, okay. I was looking at a different one. The horse, remember Equus? And you could see change over time that it got bigger and bigger? Okay then. All right. What was the next piece of evidence we used? Biogeographical. And that organisms are going to be affected by their climate of their region. Then they could point to a common ancestor, right? But you would become specific to the environment in which you live in. I would probably talk about mammals. Okay, what was the third piece of evidence we looked at?
Anatomical.
Anatomical. And under anatomical, there were 3 pieces of evidence. What were they? Homologous structures, common anatomy. We don't have to have common function. Common anatomy, which are called homologous structures. Common function is analogous, right? If you have common anatomy, you probably have common what? DNA, which means you have a common ancestor. What was the second piece of anatomical evidence? Vestigial structures, remnants of a structure once important but no longer important. It does not increase your fitness in your new environment. And what was the third piece? Embryonic development. For more similar, are you looking at your notes?
No.
That's awesome. If you're looking more similar, the embryos develop, then the more likely they are related. Okay, what was, okay, so we did fossils, we did biogeographical, and we did anatomical. What was the fourth piece? Biochemical. We have the same similar DNA and proteins, enzymes, ATP, and we looked at cytochrome C as our example, and the similarity in the primary structure of that protein. And then you could look at developmental genes as well. Hey, yeah, bye. Are you going to put your computer away? Oh, never mind.
We have a system.
Okay. We have a system. I am leaving. Okay, we reviewed all the essays, and you learned the next chapter. That was impressive.
Quick question.
Yes.
Back to the one before this, where you had those squares. Do we even need to write this square? Can we just know it's p squared plus 2p cubed plus q squared equals 1? Like, is that just different?
You use those equations, you'll support that. And they always have a Hardy-Weinberg problem on the test, somewhere.
P test?
Yeah, always. Seems like very consistent. They always have a Hardy-Weinberg.
So, in part B, like for the example, it would be P could be 25,000 over 100,000.
Well, I'm not going to work this one out for you because I want you to try it first on your own.
Okay.
Yeah, but you can work it out. Work it out. Okay, Smarties, if you're doing this really late, have a piece of toast. There we go.
Why not cake?
Or cake. You can have cake. It's just nice to have a warm piece of toast. Or you could have some pie. Have some pie.